reactions what will happen through plasma membrane. This is the most important concept where you may get confused. So I try to put all the concepts in a table. See, by observing the table itself, you can understand most of the points. Here, molecules or ions movement through plasma membrane. How it happens? By diffusion. Now, what does mean by diffusion? Diffusion is nothing but the movement of molecules down a concentration gradient. Remember the word down. So why are you using down a concentration gradient? Because molecules, when they move from high concentration to low concentration, that is called diffusion. Okay. Now, don't get confused diffusion with osmosis. Both looks the same, but they are different processes. I'll take an example and explain in the next slide. First, you better understand this table by comparing the diffusion and osmosis. Now, when we talk about diffusion, we talk only about molecules and ions. We will not specify what kind of molecules, what kind of ions, but definitely not charged ones. But when we talk about osmosis, we discuss osmosis related to water molecule only. Okay, osmosis is always related to water molecule. Remember that word first. So the movement of water molecule across, we are using a word across high water potential to low water potential. So we are saying that not down. We are using the word across. There's a slight difference in using these two words. Down means what? From high concentration to low concentration, just a flow. See, when you pour water, something like, for better example, I'm taking a crude one. When you pour water from upper surface to lower surface, do you need any pressure to push the water? No. The water will flow freely. So that's the difference between down. When we talk about across means you need some pressure to push the water molecule even though there is a concentration gradient. That is called osmosis. So when we talk about difference between diffusion and osmosis, remember always 2 points. Diffusion is related to movement of molecules. Osmosis is related to movement of water molecules. First point. Clear? Then diffusion is related to movement of molecules down the high concentration to low concentration. Whereas osmosis is movement of water molecules across high water potential to low water potential. This I'll explain by taking an example of cell, especially a plant cell or animal cell, how diffusion and osmosis happens. That I'll explain. Then we'll come to active transport. There's not much to discuss about active transport, but these 2 points you have to know the differences, because basically at your standard, the diffusion and osmosis differences would be a straight analytical question and a descriptive question. If you come to multiple choice also, they'll confuse you between these 2 processes. So I'll just explain these 2 processes by taking the hypotonic solutions and all. Okay, just a moment. I'm trying to share a slide, the same slide, but in next page. Okay. So here, when we take the diffusion process is always a simple process. Okay. Now diffusion process related to molecules and ions. What kind of molecules? It may be protein molecules, or it may be lipid molecules, or it may be simple sugar molecules. Examples I'm telling, which will move through diffusion. And ions may be calcium ions, or potassium ions, or sodium ions, something like that. Okay, so these are the ions and these are the different molecules which move through diffusion crossing plasma membrane. Remember that word. We are talking about plasma membrane structure and how different molecules are crossing the plasma membrane. So concentration gradient will be there, high concentration to low concentration, diffusion happens. Now assume that we are taking a cell. I'm taking an animal cell. Assume that this is an animal cell. Now you'll be having cytoplasm inside the cell. Now you keep the animal cell, an onion cell, a cheek cell or a liver cell, something like that. First we'll talk about animal cell, then we'll compare it plant cell. Now this is the cytoplasm and different cell organelles inside this animal cell. Now you kept it in a solution. A cheek cell example I am taking. Okay. Now you kept this cell in a solution. Now outside solution, if it is having same concentration as that of the cytoplasm, so there won't be any inflow or outflow of molecules. So no inflow, no outflow of molecules if the animal cell or plant cell is kept in a solution having same concentration as of the cytoplasm. Remember the word cytoplasm. Okay, this is first case, case 1. Now we'll see the case 2. Now in case 2, assume that the same animal cheek cell with cytoplasm and cell organelle, healthy one, is kept in a solution. But here, the solution in which the cell is kept is having high concentration when compared to the concentration of cytoplasm of the cell. This we call as hypertonic. Understand the word hyper. Hyper is nothing but high concentration. High concentration of what? High concentration of the solution in which the cell is kept. Okay. Now what will happen? As we are talking diffusion and osmosis, both the processes, what is happening? A concentration gradient will be there. Gradient is nothing but difference in concentration. It might be high concentration to low concentration movement, either down the gradient or across the partially permeable membrane, which is nothing but the plasma membrane here. Now assume that here this is the solution. Now what will happen? From high concentration to low concentration, molecules movement happens, or the water movement. Okay. So here the outside solution is having high concentration and cytoplasm is having low concentration. So what will happen? Inflow, movement of molecules into the cell happens, because of which cell swells and bursts out, ultimately dies. This is the result of case 2. So when a cell is kept in hypertonic solution, we are talking about animal cell. Remember that point. Cheek cell example I took. When animal cell is kept in hypertonic solution, a solution having high concentration than the cytoplasm, then the result will be inflow of the molecules into the cell, maybe by diffusion or by osmosis. Both the processes may happen. As a result, the cell will swell and burst out. This is case 2. Now come to case 3. Now when you take the case 3, here we keep the same cell in a solution, the same cheek cell, healthy cheek cell with cytoplasm and cell organelles in a solution having low concentration compared to the concentration of cytoplasm. This we call as hypotonic. Okay, here hypo stands for low concentration. Low concentration of the solution when compared to cytoplasm. In this case, what will happen? We are talking about high concentration to low concentration movement of molecule, isn't it? So here also there will be movement, but how? It would be outflow, isn't it? Because the outside solution is low now in concentration when compared to cytoplasm. So here the result would be outflow of cytoplasm and other cell matter into the solution. This may result in shrinking of cell and ultimately cell dies off. So in case 2 and even case 3, the animal cell, remember the word animal cell dies. In case 2, because of hypotonic, inflow of material happens, cell swells and bursts. In case of case 3, it is hypotonic, outflow of material happens and the cell shrinks and dies. Okay, so these all are examples of diffusion and osmosis, through which we can understand whether the cell is in a healthy condition or in unhealthy condition. Any doubt regarding the diffusion and osmosis processes? Of course, you know the mechanism, but we use the terms like inflow, outflow, hypertonic solution, hypotonic solution. These are the new terms, I think. Cell swelling and bursting, that you know, and cell shrinking and dying, that also you might be knowing. But the biological terms which we use here are the solution concentrations, inflow and outflow of materials.
Excuse me, ma'am.
Yes, sir. Now go ahead.
Ma'am, in hypertonic solution, the solution has higher concentration.
Yes, compared to the cytoplasm of the cell.
So, ma'am, what will happen then?
Then what happens, either by diffusion or by osmosis, there will be movement of molecules from high concentration to low concentration. So either of the mechanism may happen, and the molecules which are there in the outside solution, they'll move into the cell. That's we call inflow. Moving into the cell.
Inflow will be greater than outflow.
Outflow, we cannot compare outflow here. Outflow will be there only when it is hypotonic. See, inflow and outflow we are using based on the permeability nature of glass membrane. There will be always movement of molecules, but we won't call that as inflow and outflow. Okay, got it?
Yes, ma'am.
So when the molecules comes inside, what will happen? The cell will swell and burst out, isn't it? So the in hypotonic solution, the quite reverse happens. So outflow happens. The more content of the cell will move out because of the osmosis and diffusion. So that results in shrinking and withering of the animal cell, and it may die off. So this we can talk in terms of cheek cells as if example I gave, but usually they'll ask about RBC. We'll see. I took some exercise questions. We'll see how that also will be justified in multiple choice question pattern. Okay, Bhumika, do you have any doubt? I think you just off and on. I mean, do you have any doubt regarding diffusion and osmosis?
No, ma'am.
Okay, fine. Then active transport. Active transport is nothing but just a moment. Here. When you take active transport is related to substances. See, why I chose this slide, if you are taking notes, try to copy this, because this whole chart explains various examples. See, diffusion related to what kind of molecules movement. Osmosis related to what kind of molecules. Water molecules only, osmosis. Then when we take active transport, it's nothing but movement of large molecules. In the earlier slide I showed some charged molecules we talked about, isn't it? They are not easily moving into the cell through the glass membrane. So in such condition, what we need is, we need energy. Energy is required for the movement of charged molecules across the plasma membrane. So that is called active transport. The quite opposite one is called passive transport, where no energy is required. So this is one method of movement of molecules along with diffusion and osmosis. So when we talk about the movement of molecules or ions or water molecules or any charged molecules through the glass membrane, we'll have basically 4 methods. I mean 4 processes we can say. One is diffusion, other one is osmosis, The one is active transport, which requires energy for the transport of molecules or substances. And the fourth one is passive transport. This is also movement of molecules, the charged molecules across plasma membrane, but it won't require energy. So it doesn't carry much importance. That's why I didn't include it in this chart. But these three carries a lot more significance when we talk about functional aspect of plasma membrane. Clear? Any doubt about diffusion, osmosis, active transport? If it is clear, we can move on to the next one.
Ma'am, can you once again explain active transport?
Active transport is just a process in which the charged ions or the charged particles will move from outside environment into the cell through plasma membrane. Okay, that's the active transport is related to charged ions and particles movement across plasma membrane. This is the first point. And the second point is the active transport movement of molecules requires energy for the transport of substances outside cell to inside cell. Requirement of energy is the basic point behind active transport. Okay.
Okay, ma'am.
Okay. Then we have the most significant feature of a plasma membrane,